Human brains have billions of neurons working together to precisely coordinate every thought and action from our minds to our bodies. Over time, these connections may start to degrade as the body ages. While these changes in our brains often start slow, forgetting small things here and there, eventually the change becomes noticeable as cognitive decline progresses. This progressive loss of neurons is known as neurodegeneration, which is associated with deposition of harmful proteins in the brain and synaptic dysfunction. This is a healthy neuron. Here we see the dendrites, which are responsible for receiving and processing signals from the axon terminals of other neurons. The soma is the cell body of the neuron and contains the nucleus. The axon is lined with myelin and helps axons conduct electrical signals to the axon terminal. Here, an excitatory or inhibitory message is sent to the next neuron. This forms networks of neurons that work together to relay signals at the synapse, a point between the dendrite and axon terminal of different neurons. The synapse is where neurons can release charged particles from presynaptic axon terminals to postsynaptic dendrites. In a healthy individual, neurons send signals using action potentials. Action potentials are electrical impulses that allow neurons to communicate by causing shifts in the neuron's potential energy through binding of neurotransmitters or electrolytes. Neurotransmitters can trigger a response in the postsynaptic neuron, causing the propagation of an action potential or the inhibition of one. Neurodegenerative diseases limit the brain's abilities to communicate with the body by disrupting synaptic signaling. The most common of these disorders include Alzheimer's and Huntington's disease, affecting both an individual's mood, speech, balance, and memory. About 1 in 9 people age 65 and older have Alzheimer's dementia. That is 7.2 million people in the United States, not including those with early onset. Early onset dementia can start as young as age 30, usually in people who have gene mutations causing amyloid structures to form. Amyloid structures are clusters of highly ordered proteins that disrupt biological function. Disruptions in the brain can cause disorders such as Alzheimer's and Huntington's disease, affecting an individual's cognitive ability. Today, most Americans have a loved one with a neurodegenerative disorder, highlighting the importance of research in this field. Alzheimer's is a neurodegenerative disorder considered a disease of synaptic failure due to beta amyloid structures. Along with beta amyloid deposition, tau hyperphosphorylation also disrupts the synapse, the location where information is transferred between neurons. Under normal conditions, amyloid precursor protein is typically cleaved by alpha secretase into non-toxic peptides. Tau, located primarily in axons, supports axonal transport. In the disease state, amyloid precursor protein is processed through the amyloidogenic pathway, where beta secretase and gamma secretase generate these beta amyloid peptides that are prone to aggregation. Beta amyloid peptides are produced by the cleavage of amyloid precursor protein. While beta amyloid monomers are soluble, They can aggregate to form insoluble oligomers and fibrils that are the primary component of amyloid plaques. Here we see the path of beta amyloid monomers clumping together to form larger molecules like oligomers. The oligomers come together forming beta amyloid fibers, forming beta amyloid plaques. While these plaques are formed, it induces an inflammatory response, triggering the formation of tau, which leads to the conversion of healthy neurons to diseased neurons. The diseased neurons trigger inflammation in the brain, which in turn causes more neuron loss, leading to cognitive decline, similar to a negative feedback loop. Here we see an image of the 3D structure of amyloid fibrils, discovered through the use of solid-state nuclear magnetic resonance, NMR. The 3D structure is composed of parallel beta strands linked by short loops. The interactions between the three strands are hydrophobic, and the surface residues are mostly charged, forming several salt bridges. The three-stranded triangular hydrophobic core superimposes well with the beta structures of soluble proteins. These structural arrangements result in an overall beta helix characteristic fold. These beta amyloid fibrils disrupt synaptic plasticity and mediate synaptic toxicity through multiple mechanisms, including interference with glutamic receptor signaling, its cycle, and display synergistic toxic effects in combination with tau. Beta amyloid structures disrupt synaptic plasticity by acting on glutamate receptors that mediate action potentials in the brain. It does this through activating NMDARs, a subtype of glutamate receptors, allowing calcium into the brain. This overload suppresses LTP, a cellular mechanism for learning and memory. Calcium then activates the kinase CAMK2, leading to synaptic loss through destabilization of AMPARs, a glutamate receptor involved in excitatory synaptic transmission. Beta amyloid plaques also disrupt glutamate homeostasis and enhance excitotoxic signaling through promoting glutamate aggregation. The plaques cause an increase in glutamate spillover through nicotinic acetylcholine receptors, which excessively activates NMDARs, resulting in increased calcium levels. Overproduction of beta amyloid oligomers are mediated by endocytosis of NMDARs, further increasing the glutamate aggregation as it decreases glutamate uptake. Beta amyloids can activate astrocytes, which release pro-inflammatory cytokines, causing more beta amyloid aggregates to form. Beta amyloid fibrils also disrupt synaptic plasticity through synergistic effects in combination with tau. Tau is a protein that functions in regulating microtubule assembly, axonal transport, and stabilization. Under pathological conditions, tau's affinity to microtubules lessens, causing it to detach from axonal microtubules and form aggregates. Beta amyloid structures increase the activity of kinases such as GSK3 beta, and CDK5, which promote tau hyperphosphorylation. This leads to inhibition of LTP and cognitive impairment. Phosphorylation of tau proteins can mediate FYN kinase facilitation to dendrites interacting with a PSD95 NMDAR complex, inducing synaptic toxicity. In a healthy brain, tau helps reduce oxidative damage. In Alzheimer's disease, tau becomes excessively phosphorylated by kinases, causing it to misfold and aggregate into tangles. This causes tau to detach from axonal microtubules, impairing the integrity of microtubules and inducing synaptic dysfunction. Phosphorylated tau is more often found in the hippocampus and accumulates at synaptic terminals. These molecular changes can be caused by neuroinflammation, driving synaptic dysfunction and neuronal loss. It is important to note tau's effect on neuronal synapses is not fully understood just yet. The impairment of synaptic signaling means neuronal cells can no longer properly communicate between different parts of the brain. An example of this that I've observed while working with patients at an assisted living is when someone knows what they want to say, but they cannot verbally express it. Their brain is unable to communicate their thoughts into spoken words. This is extremely frustrating for patients, and sometimes having them write on paper can help, as it uses different signaling pathways in the brain. Like Alzheimer's, Huntington's disease is another neurodegenerative disorder resulting in motor dysfunction and cognitive decline. Neuronal degeneration mainly occurs in spiny projection neurons in the striatum. This location is critical for voluntary and involuntary movement, in which Huntington's disease disrupts these neural pathways. Huntington's disease is caused by a mutation producing CAG trinucleotide repeats in the Huntington gene, resulting in the pathogenic mutant HTT protein. Non-diseased Huntington gene acts as a scaffolding protein, functioning to stabilize intracellular molecules into motors, allowing fast axonal transport. This neurodegenerative disorder causes deficits in synaptic plasticity. Patients with Huntington's express more mutant forms of the gene rather than the non-pathogenic form. Reduction of the Huntington gene negatively impacts synaptic plasticity in the brain through decreasing LTP. LTP is crucial for synaptic strength, occurring when neurons repeatedly activate, building the basis for synaptic learning and memory. Non-mutagenic Huntington disease upregulates the synthesis of a neurotrophin called BDNF, which enhances LTP. So the decrease of non-pathogenic Huntington gene in patients leads to a decrease in BDNF synthesis, impairing LTP. So in short, we want the non-mutagenic form of the gene. In recent years, research has looked into ADAM10, alpha secretase, which interacts with the Huntington gene and is involved in presynaptic homeostasis. ADAM10 cleaves amyloid precursor protein in a non-amyloidogenic fashion, leading to less beta amyloid plaques. While Alzheimer's disease has reduced ADAM10 levels, it is hyperactive in the brains of Huntington's. Although beta amyloid aggregates are not a major point of overlap between Alzheimer's and Huntington's disease, the involvement of tau protein displays molecular similarity between the two disorders. In patients, the tau gene, MAPT, has been associated with Huntington's disease. Experiments have been investigating the role of MAPT in model organisms to get a better understanding on how tau impacts the brain. Knockouts of the diseased tau protein in mouse models were found to improve motor functions. Studies on postmortem diseased brain samples have found increases in phosphorylated tau protein levels in patients with late stages of the disease. Postmortem studies in early onset patients suggest a correlation of tau pathology with disease progression instead of it being prompted by age. The negative impacts of Alzheimer's and Huntington's disease is still being investigated. While progress has been made in treatments, a cure for either disease has not been discovered. Currently, monoclonal antibodies have been showing progress in treatment for Alzheimer's patients with beta amyloid pathology. Anti-amyloid monoclonal antibodies, referred to as MABs or MABs, all target high molecular weights of beta amyloid structures. Medications using monoclonal antibodies differ in infusion frequency and half-life, allowing patients to have treatment options that best suit their needs. Here we see how anti-amyloid monoclonal antibodies work to target beta amyloid aggregates. The anti-amyloid monoclonal antibodies activate microglia, the brain's immune cells. The monoclonal antibodies are designed to target epitopes that match the amyloid structures and bind to it, signaling microglia to target those structures, ingesting them through the use of phagocytosis. When the plaques have been ingested, they are degraded by lysosomes and endosomes. In terms of treatment for tau hyperphosphorylation, kinase inhibitors may prove effective as it would prevent tau from being phosphorylated. Currently, immunotherapies for targeting tau proteins are in the clinical trial stages. Treatment for Huntington's disease also shows promising results from antibodies using mice. Researchers use an anti-tau antibody, finding a decrease in hippocampal tau protein. The mice that got antibody treatment showed improvements in motor function and cognitive ability. Another potential treatment for Huntington's is RNA targeting of the mutant Huntington gene, which has recently entered clinical trial stages. This method is thought to be able to decrease the mutant Huntington gene production by using RNAi to silence the gene expression. While medications can help loved ones as cognitive abilities decline, outside help may be necessary. I have worked the last 4 years with people affected by cognitive impairment in both assisted livings and the patient's home. I believe it is important to note how these diseases do not affect everyone the same. Huntington's and Alzheimer's disease are a spectrum, with no two people being alike. So it is important not to characterize them based off prior experiences or stereotypes. These are unique individuals who are going through the most difficult times of their lives and need to be treated with compassion. I want to note that just because someone has the disease doesn't mean they can't form new connections or live fulfilling lives. At the assisted living I work at, we refer to the patients as residents. The people living there are able to make friends and enjoy various activities, including playing sports and cooking. The residents frequently leave the facility through organized outings to restaurants, sporting events, and nature hikes. Many of the residents have also gone to concerts with friends that they have met at the facility, and they're still able to take care of their pets by going to vet visits. In our current age with cell phones, many of them still have their own private phone and are able to call their loved ones without assistance. The cost of an assisted living can range quite drastically. The place that I work at costs an exorbitant amount that most individuals unfortunately cannot afford. For those not requiring around the clock assistance, in home treatment is also available, also as a more affordable option. I work as a companion to those who are able to maintain themselves but need light assistance. This offers families peace of mind that their loved one is safe while allowing them freedom and the ability to stay in their own home where they are comfortable. I hope that moving forward, treatment options for these disorders become more affordable. With current clinical trials, medications have the potential to advance our knowledge and offer better quality of life opportunities. This research project is dedicated to everyone affected by neurodegenerative disorders, to those who struggle to live with the disease, the families and caregivers who fight to advocate and support them, and the researchers working to make critical advancements. All right, we have reached the end of the video. I want to lastly thank everyone that I've worked with over the last 4 years. It has meant the world to me to help make positive changes in people's lives. And I would like to thank all of my sources because there's so much information on here. So thank you to all of those researchers. And thank you to everyone who has watched this video. I have poured my heart and soul into the last 25 to 30 hours of work to make this 20-minute long video. So thank you all for watching.